Okay, being totally honest, I absolutely forgot this whole section when we started this video, so it's going to feel disjointed because I started in the wrong spot. But there's a couple of things that we need to know. So our essential question on this part before we get into the next part are what are variations on Mendel's law? Variation on Mendel's law. So most things are not going to fit into Mendel's pattern of inheritance. So his P crosses always looked like two parental varieties, and so that was what we call complete dominance. But for some characters, or actually a lot of characters, the appearance of the F1 of the first sons and daughters fall between the phenotypes of the two parental varieties. So this is called incomplete dominance. And I say most of the time for incomplete dominance, it looks like blending. You typically see them sort of blended together. So an example is the snapdragon flower colors. So we have kind of big R is the red, little r is the white, but if we have the hybrids or the heterozygotes, they come out pink. So we have an extra phenotype that's there. So an extra phenotype is present. One example of incomplete dominance is human is called hypercholesterolemia. It just means the dangerously high levels of cholesterol occur in the blood. So heterozygotes have this kind of intermediate high levels. And so a normal cell has plenty of these little LDL receptors. Severe disease has none. And if you're heterozygous, you'll appear sort of in the middle, where you've got some and not all of them. So it's sort of like a blend of these two. Kind of hard to imagine a blend of like cell receptors, but that's the best we get. Although each individual carries at most two different alleles for a particular gene, in cases of multiple alleles, more than two possible alleles exist in a population. Where do we get this example? Blood type. So human ABO blood phenotypes involve three alleles for a single gene. So four human blood types A, B, and O result from combinations of three alleles. A and B alleles are both expressed in the heterozygous individuals, so we call them codominant because they both dominate certain areas of the cell, but they have their kind of areas. So I will say for a lot of codominance, you'll see it sort of be more patchy, less of like a blend. So we can talk about this. Blood type is fascinating, and it gives you the I on that one, which is strange, but it's telling you the locus or the particular location of that gene. So A may have genotypes of big A, and then there's big A, and then there's this little I, which just says the recessive. Same with B, big B, and then the I, little I. AB is this codominant A and B, and then for O, it's little I, little I. But the thing to look at is for A, is that it will express with these little markers here, like little triangles, as the antigen for carbohydrate A. B will show as like little circles, and if we have AB, it will have both triangles and circles that are represented. And then for O, the recessive, none. This is an example, too, where the recessive is actually the most common in the population. So technically, if we went to those terms, O would be the wild type. There's a fun fact that we'll look at is that blood types don't get along with, like, if it's an A, it really conflicts with B. So an A person can only get blood from O or an A because they have the same antigen types. Even though there's the little triangles represented here, the fact that there's circles really makes the A blood angry, and it won't recognize it as self, so it will try to destroy it. Same with B, is that if there's A present at all, it will attempt to destroy it, so it can only take O and B. If AB, these are what we call the universal acceptor, is they can take blood from anybody because they have all the things that recognize all the self. So it's like, yeah, let's put anything in there. O, on the other hand, is the universal donor. As you can see, everybody can take B because they recognize it as self. It doesn't have any conflicting antigens. But Lord knows if you put A or B in there, it's like, ooh, there's nothing here. So we can talk more about that, but that is a little side note on how blood type works. You should ask your parents, see if they know what your blood type is. Mine is O. I'm O minus. Pleiotropy. So here's a new thing. Pleiotropy occurs when one gene influences multiple characteristics. Sickle cell is an example of that. So it affects the hemoglobin produced. So it takes a nice pretty cell and then makes it kind of sickle shaped. And in doing so, it'll actually cause anemia, organ damage, like blood clots, all that kind of stuff, which means that we're getting more than one influences. But we actually keep sickle cell around in some African countries because of its resistance to malaria. So there's an evolutionary advantage of it being there, because malaria will not hang out with a cell that's that shaped. Ain't doing it. Okay, so here's what we kind of look for pleiotropy. So individual homozygous for the sickle cell allele produces sickle cell of normal hemoglobin, then it becomes sickle shaped, then we have damage to organs and other effects. All of these things just from one tiny allele change. It's a big deal. It's actually one single base pair, so we call it a point mutation, creates all these effects. Here's another one. So many characters result from what we call polygenic inheritance. So of course that's many genes. So many genes actually influence a single character. So a single phenotypic character results from the additive effects of two or more genes. So many genes. Human skin color is an example of that. Most of those things where you get a range, like from very light to very dark, you can think even, I think eye color is as well as polygenic. I think even hair color, height, anything where you get a range is usually polygenic. And, you know, you'd be wrong on that one, but you can think of kind of a range of things. Many characters result from a combination of heredity and environment. So your skin color is affected by exposure to sunlight. Heart disease and cancer are influenced by genes in the environment. Identical twins can show that a parent's traits are results of genetic and the environment. That's a fascinating one, because they should have the same, but we've got a range of skin colors and all kinds of things, and as you increase melanin, as you get sunlight exposure, so we'll do sunlight exposure to all of them. You should go to all of them. There we go. As you expose to sunlight, that can change the amount of skin color, so there's an environmental component as well as a genetic component. Same with hair color. Okay, and these identical twins, they've got their own kind of thing going on, and so that leads to some phenotypic or expressed differences. Okay, I finally made it back to where you're supposed to start to, so it's going to like erase all this and start over, but my bad. Anyways, keep it going. In today's lecture, we're going to look at the chromosomal basis for inheritance. So our essential question today can just be, what is the chromosomal basis for inheritance? So, basis for inheritance. I don't have a stylus today, so this is just my hand. But I want to also mention that these are things that just kind of don't follow Mendel's laws. So I'll put don't follow Mendel's, because that's basically what we're looking at. So the chromosomal theory of inheritance states that genes occupy specific loci or loci or positions on chromosomes and will undergo segregation and independent assortment during meiosis. So these are two important ones to look at, is that segregation and independent assortment. Those are coming up. So Mendel's law correlate with chromosome separation and meiosis. So the law of segregation, and this part's important, it states that pairs of alleles separate from each other during gamete formation via meiosis. So whenever we split things into four, we're saying that each kind of each allele, so each variation of the gene that we have, which we should have two of them, will separate from each other whenever we go from, remember, diploid to haploid. So whenever we go to haploid, those two N are going to separate from each other. And it depends on the separation of homologous chromosomes in anaphase 1. The other thing that's important in this section is the law of independent assortment. It states that each pair of alleles sorts independently of other pairs of alleles during gamete formation. So basically, this one's about alleles, and this one's about different genes. So it says that different genes don't depend on each other. Okay. So if this one's alleles, it's like for one particular thing, if your mom gave you a copy for blue eyes and your dad gave you something for green eyes, well then those two will have a separate chance of going into the gametes in the future. Law of independent assortment just says that basically like eye color and like having a widow's peak, those things don't depend on each other. So they're independent of each other. So different genes are independent of each other. So what it shows us here is that in the F1 generation, in our first sons and daughters, maybe we have all these round and yellow seeds. And so we see 4 different chromosomes, and the homologous chromosomes line up once they're paired. Okay, in anaphase 1, those homologous pairs will separate, and so that's kind of at this phase is where we see the law of segregation. And put law of seg for short. But when we enter metaphase 2, the whole point is that these guys can line up differently. So this is one possibility where these guys had lined up in the middle, where kind of all the red ones were to the left and all the blue ones were to the right. However, we could have the possibility where blue and red have different mixtures. And I think you guys saw this when you were doing your actual meiosis models. You could kind of see that you could line it up differently. And that would help us say independent assortment is that the genes on these different chromosomes don't depend on each other. So just because blue went this way this time doesn't mean that also this blue must come with it. It means that we can change them up. Okay, this is going to be a beast, and we'll go over some of these together as we try it, but this is what's called a dihybrid cross. And, you know, as much as I always gripe about the state of Texas, they do love to put a dihybrid cross on there. We could talk about it, but it's just better to practice it. So we're going to go through it in class a little bit better. So you can sort of look at it and get an idea, and I'll show you what's going on here, but it's a mess. It's just better to try together. Okay, so Bateson and Punnett studied plants that did not show a 9:3 to 3:1 ratio. That's phenotypic stuff. So it's saying that of these 16 in this dihybrid cross, 9 of them have both dominant of being yellow and round. 1 have both recessive of being green and wrinkled. And then we saw these kinds of variations where you could see 1 dominant and 1 recessive. So 3 showed dominant for yellow, recessive for wrinkled. And then 3 showed the recessive for green, but dominant for round. So that kind of says that, you know, we can have some mixtures in there whenever we're trying. This is actually for 2 different genes. Excuse me. Should have explained that. So this says this for different genes. And we have 2 different genes that we're crossing at the same time. So that's where the dihybrid cross comes from. I don't know why my screen's doing all kinds of weird righty things, so it probably I need to clean it. Anywho, so he actually studied some. So 9:3 to 3:1 would be what we would expect in the F2s of the second sons and daughters if everything was working out right. But when it doesn't work out right and it's not perfect, which is most of life, we can actually find things called linked genes. These are worth writing down. So linked genes are located close together on the same chromosome. So these 2 are going to travel together. Instead of being independently assorting, they're going to travel together because they're on the same gene. So them being on the same gene means they tend to be inherited together. All right, so how do we have this one? So if this one has an experiment where we have a purple flower and long pollen, we would expect to see a 9:3 to 3:1 ratio here. but this is what we expect, but what we're actually seeing is more of the purple and long. So more showing the dominant and actually more showing this kind of long and round here. Only a few of these kind of middles where we've switched things that have kind of half dominant, half recessive sort of traits. So what that says is that these genes are linked together on the chromosome. That's kind of all I'll go with on that one. It's actually when they're linked in this way, they tend to show more of the 3-to-1 ratio, which is sort of what we're getting at. However, it doesn't work perfectly, as you can see kind of before, is we did get some with the cross of each, but just not as many. And why would we still get them? Because of crossing over. So also go back to thinking of your meiosis models where we crossed over little pieces. So when we cross over homologous chromosomes, so you get like different colors, maybe green and yellow on your chromosomes, the crossing over actually does create new combination of alleles and gametes. So that would sort of mimic the independent assortment. It just doesn't do it as often. Okay, so linked genes can be separated by crossing over, forming recombinant gametes. So the percentage of recombinant offspring is called the recombination frequency. All right, so just we have some, and again during crossing over they switch little pieces, and you notice we've got a little recombinant here. So we've switched it up from those parental gametes. We do this in Drosophila a lot. So this is Drosophila melanogaster, which is a lovely little fruit fly. We mess with its poor genetics all the time. So they did an experiment kind of with the body color and these kind of vestigial wings just to see what would happen. There's a lot happening here, which I'm not going to get into all the details. But the whole point of that is that with Sturdivant, there was a guy, Sturdivant, that found that the greater the distance between the two genes on a chromosome, the more points there are between them where crossing over can occur. So that might be worth noting to kind of figure that out. So recombination frequencies can thus be used to map the relative positions of genes. So we can actually kind of create a map of where these linked genes fit. So we call that a linkage map, a map of where linked genes fit. So just to kind of give you an idea, if we see that 17% of the time this black band and this blue band cross over 17% of the time, that just tells us that they're a little bit further apart. And they're representing, again, these are loci, so these are representing the location of different genes. But if we notice that black and gold recombine 9% of the time and gold and blue about 9% of the time, we can sort of make some assumptions that the gold sits in between the two, just to kind of get an idea. So here's some that they're able to figure out the mutant phenotypes or what's going on with these guys. So they're able to figure out, they have long or short antennae versus body color versus type of eye color versus some wing issues versus another set of eye colors. So they're actually able to map these things by how often they cross over. So we would assume that these kind of long sort of antennae and red eyes, will assume that they'll cross over more often because of how far away they are, and that maybe red eyes here and normal wings will cross over less times because of how close they are. So that's all I wanted to give you on that one. This is a great place. We're going to talk about sex chromosomes and sex-linked genes, but we're at the 10-minute mark, so this might be a great time for you to pause, take a breather, review your notes, and then come back. Try to actually pause this time. You guys are crazy. All right, so many animals have a pair of sex chromosomes. So we kind of talked about that already. And I realized, I guess I got ahead of us on that before, but just in case, to review that one more time, we're designated X and Y, and they determine an individual's sex. So in particularly humans, and there's some other mammals, individuals with one X and one Y are males, and two X's are females. So especially if you didn't get that before, that's definitely important to know. In addition, human males and females both have 44 autosomes or non-sex chromosomes. So that's essentially pairs 1 through 22. whereas pair 23 is the sex chromosomes. So pairs 1 through 22 equals 44, non-sex or autosomal, self-chromosomes, non-sex chromosomes, and pair 23 is the sex chromosome. So in mammals, including humans, the Y chromosome actually has a crucial gene called SRY for the development of testes. So the absence of the SRY gene directs ovaries to develop. And that's kind of how I'd mentioned before. So Y really, there are a few other genes that exist and loci that are there, but it is fairly reduced in size as far as the chromosome goes. But it does have the crucial gene of SRY, which develops testes. Okay, so you can see these in comparison. The X chromosome is a little bit larger, and the Y chromosome is fairly small. So just to show, we've got diploid or diploid parents. This is a male with XY and a female with XX, so that 23rd pair is the X and Y. In gametes, they'll be haploid with 22 chromosomes plus an X and a Y, because remember, law of segregation, equal chance of X and Y's going into the next set. Females only have X's, so it's going to be 22 plus an X that goes into every egg, because we've only got one choice there. And so the offspring will either be female or male, depending on how those work when we go back to being diploid or diploid after fertilization, for short. Okay, so kind of a way to review that. What's interesting, though, is not everything operates off of this system. So some of my favorite things, like grasshoppers and roaches and insects, have what's called an XO system, in which O stands for the absence of a sex chromosome. So females are XX and males are XO, so they only have one copy of X. By the way, I don't think it's really asked all that often. It's just fascinating stuff, so you don't need to write too much here. In certain fish, butterflies, and birds, the chromosomes are Z and W. So males are ZZ and females are ZW. So the difference there is that the males are homozygous for the sex chromosomes, or not, I guess not homozygous. That it's kind of the same. You could still think of it the same way. Some organisms lack sex chromosomes altogether. So most ants and bees, sex is determined by chromosome number. This is fascinating to me. So females develop from fertilized eggs and males develop from unfertilized eggs. So the whole point is that the females will be diploid and the males will be haploid. It's crazy. They also don't have fathers. So it makes this whole thing nutty. Like all the females are 3/4 related to all the offspring. Yeah, that's why they have more investment. The females are the only ones that sting, but they have that investment. So again, XO system, males 1X, females 2X's, ZW, male ZZ, female ZW. And you can see there are more chromosomes in chickens. And then chromosome number for bees is 16 for males, 32 for females. So they're a little less, but interesting nonetheless. In some animals, environmental temperature actually determines the sex. So a lot with reptiles, the temperature the eggs are incubated at during a specific period of embryonic development determines whether it's male or female. So it's about temperature for these guys, which does bring about the question of global climate change. So if we alter or change the climate because of CO2 emissions or whatever, it actually can affect sex ratio. So maybe more males will be developed than females, or more females than males. So it impacts our kind of ratio for genders of those particular babies. All right, now to some important stuff. I mean, it's all kind of important and fascinating, but this part's what we really need. So we have sex-linked genes, and they're located on either of the sex chromosomes. So if we have a gene that's on a sex chromosomes, it is sex-linked. So the X chromosome carries many genes unrelated to sex. So the inheritance of white eye color in the fruit fly illustrates a sex-linked or X-linked recessive trait. So it's sex-linked, so it's on the sex chromosome, the white is. So if we have a female that is heterozygous or homozygous dominant for red, and a male that has one copy of the recessive trait, he actually only needs one copy here. Why? Because there's no dominant to mask it. So that's where the tricky part gets in here, is that if a male has just one copy of the recessive, it will express because there's nothing else there to mask it on the Y. So when we do the crosses, we'll get a sperm from the male, which will be the little r and the Y, and then the female will have two X with the big R's, so it's just giving us it's telling us it's on the sex chromosome of X. That's why there's the big X in front of it. And then afterwards, we have a homozygous for the dominant of red eyes. And so no matter what, this particular baby will express the dominant R, and it's female. And this one will be male and will express the dominant as well. So they both have red eyes. Okay, females need two copies of things to express the recessive, which is why it won't show up as much. So the next thing that we have here is another example. So if we have both female and male, and they both exhibit the red, but notice the female here is a carrier of that recessive trait, so she still has the recessive there. She would still demonstrate or show the phenotype would be red, but she does have the little recessive allele present. This particular male is normal and has the red eyes present. However, when they have offspring, and they separate, when we have the big R, little r of the female there, we'll get one daughter that's normal and the next daughter that's normal that still has the recessive there. But when we look at the sons, one half of the sons will be fine and the other half would get the recessive from the mother because of the way that those go together. So we could say half female, half males, but of the males, their chances of getting the recessive characteristic are half the time. So sometimes you have to look at the male and the female situation. Okay. So if in this case also we have a male with the recessive characteristic there, and so he shows the white eyes, again, the female is still a carrier. What can happen there is then we finally get some daughters that have recessive, but this only happens when we have a carrier and a male that's recessive. So only when we have this kind of situation going can we get females that are recessive. Again, the same with the male here. But it takes a little bit more for females to actually have that trait. So most sex-linked human disorders are due to recessive alleles and seen mostly in males. Because as we showed with the last one, it actually takes the female being a carrier and the male exhibiting the trait for those things to work together. So a male receiving a single X-linked recessive allele from his mother will have the disorder. A female must receive the allele from both parents to be affected. That's why we see most of these things in males. So red-green color blindness, for example. Another example, hemophilia, which is a terrible name. It's just excessive bleeding. So the actual name means to love blood, which is terrible. But it's where they lack more than one proteins for blood clotting. And then there's one called Duchenne muscular dystrophy, and so it's characterized by progressive weakening of the muscles and loss of coordination. Pretty sad. But the biggest one is kind of red-green color blindness. I'll also say I think alopecia or like baldness is sex-linked as well, if I understand correctly. So we can look at these things a little bit later on too, but they give us the carrier, they give us who's affected. Males are the squares here for when it comes to pedigrees. So males are always squares, it's terrible. But we had a normal male and Queen Victoria was a carrier. So her son Louis was fine, Alice was a carrier, not a big deal. Alexandra was fine, and she married Nicholas II of Russia, and so when they had a baby, they actually had a son, Alexis, who did inherit his mother's carrying trait. This is also one of those things about why it's not a good idea to do incest, for example. Keeping those things running in families does tend to amplify the recessive traits. Now we can take a little break at this point if you need a break, and then come back. Make sure to chunk your notes, take a look at them, write a few questions, start that summary. All right, so the Y chromosome provides clues about the human male evolution because the Y chromosomes are passed intact from father to son. The only way that a son can get the Y chromosome is from his father. So mutations in the Y chromosome reveal data about recent shared ancestry. So just some interesting facts here that in 2003, geneticists discovered that 8% of the males currently living in Central Asia have Y chromosomes of striking genetic similarity, which almost seems terrifying, but when they further analysis traced their common genetic heritage to a single man living about 1,000 years ago. So the combination with historical records and data led to the speculation that the Mongolian ruler Genghis Khan may be responsible for the spread of telltale chromosome nearly 16 million men living today share. There's Genghis Khan. Historically, he was not a very nice dude. Learned this. I was talking to some history teachers about this stuff, and it's like, he was kind of a jerk. Something to look into. I think that's where, yeah. So that's actually where we finish on this particular lecture. Yeah, that's a good place to stop at this one. So here's this one. If you have any questions at all, please feel free to shoot me an email. Let's make sure that we get it. Lots of things happening here, and it'll be good.